The cell cycle is the series of events that takes place within the cell before it goes into cell division. It begins in the gap phase, or GO. This is a temporary resting phase where the cell is not dividing. Then it moves into gap 1, or G1 phase. Here the cell grows and the G1 checkpoint tells the cell if it is ready for the next phase. During S phase, DNA replication occurs. G2 is the last phase before mitosis. The cell continues to grow and the G2 checkpoint makes sure that the cell is prepared for mitosis. Phases GO through G2 are grouped together as interphase. The cell spends most of its life here. M phase is mitosis. The cell will go through interphase, prophase, metaphase, anaphase, telophase, and then cytokinesis. Mitosis is the shortest part of the cell cycle. The cell starts out in interphase, where the DNA replicates. Two pairs of centrioles lie just outside the nucleus. In prophase, the DNA condenses into chromosomes. Spindle fibers begin to form from the centrioles and the nuclear membrane dissolves. During metaphase, the chromosomes line up in the middle of the cell. The spindle fibers attach to the chromosomes at a point called the kinetochore. Anaphase begins and the chromatids move to opposite ends of the cell due to the shortening of the spindle fibers. In telophase, a cleavage furrow begins to form, initiating the division into two separate cells. New nuclear membranes form around the separated chromosomes. Cytokinesis is the last stage of cell division. Here, the cell completely divides into two separate cells. Interphase of meiosis 1 is exactly the same as it is in mitosis. The DNA replicates and two pairs of centrioles lie just outside the nucleus. Just like in prophase of mitosis, the DNA condenses into chromosomes. Spindle fibers begin to form from the centrioles and the nuclear membrane dissolves. The only difference here is that this is where crossing over occurs. This time during metaphase, the chromosome pairs line up on the opposite sides of the metaphase plate. Again, the spindle fibers attach at the kinetochore. Here in anaphase 1, the chromosome pairs separate, not the sister chromatids. As in telophase of mitosis, a cleavage furrow begins to form, initiating the division into two separate daughter cells. New nuclear membranes form around the separated chromosomes. Cytokinesis of meiosis 1 is exactly the same as it is in mitosis. The cell completely divides. Interphase 2 is different from interphase 1 and interphase of mitosis in that the DNA does not replicate in this phase. Prophase 2 is exactly the same as prophase 1, except that this time, crossing over does not occur. Metaphase 2 is similar to metaphase of mitosis. The chromosomes align along the metaphase plate. Anaphase 2 begins and the chromatids separate due to the shortening of the spindle fibers. Telophase 2 is exactly the same as telophase 1 and telophase of mitosis. A cleavage furrow forms and the cells begin to divide. As in previous phases of cytokinesis, the cell completely divides. If you love